In this video we will discuss about another properties of the crowding, that is impact of crowding on cell surface association and exchange of enzyme. So they did two different kind of experiment to study this cell surface association and exchange of enzyme. The first studies map is first studies overview is shown in the panel A, where what they did is they take a cell surface layer and first they put it with this transparent layer, put a transparent layer on this cell surface, and thereafter they uh, keep these GFP enzymes, GFP fluorescent enzymes on this transparent layer, and then observe this fluorescence at different time interval to check the diffusion rate. So this is their first experiments that they have designed in order to understand the impact of crowding on cell surface association. So when they did with PBS, and after 2 minutes they are able to see some green fluorescence image. And for the Dextran, when they did it, they can see the at 180 milligram per mg they can see some fluorescence image. And after that, for this Dextran at 320 milligram per ml, they see a very low fluorescence image after 2 minutes, indicating that with increase in the concentration of Dextran, the diffusion goes down. And after some times, after 7 minutes, they can see a more high fluorescence image for the 180 milligram per ml concentration of Dextran. And but it is but for this 320 milligram per ml Dextran, the amount of fluorescence or the intensity of fluorescence is low, and it is increased with time. And these things are quantified in the panel C, where we can see that for the PBS the diffusion rate are usually highest, and after that it goes high. After that the diffusion rates are high for this 180 milligram per ml, and it is the lowest for this 320 milligram per ml, indicating that with increase in the concentrations, the with increase in the concentration of Crowder molecular, the diffusion rate goes down. Thereafter, they want to check the exchange of enzymes, impact of crowding on the exchange of enzymes. So in this case, they took a cell surface, and this time they directly put these GFP enzymes and then put another enzyme, that is CFP enzymes, and check the diffusion of the CFP enzymes into the GFP. And for the PBS, we can see that after 5 minutes, all the layers will be covered with this CFP, indicating that the exchange of enzymes is highest for the PBS. But for the Dextran at different concentrations, at 180 milligram per concentrations, we can see very low CFP formations, and even after 60 minutes, we can see most of the surface is covered by CFP, and at 95 minutes, there is a further increase in the surface covered by CFP. But for this exchange of enzymes is even slower for this 20 milligram per ml concentration of Dextran, where we can see that some of the CFP are shown at 95 minutes, but till 60 minutes, almost all the layer is covered with GFP. And these things are quantified in the panel F, where we can see that the GFP enzymes on the cell surface are a percentage of GFP enzymes on the cell surface are decreased just after 20 minutes tremendously for the PBS control, and they will keep on continue for a longer duration of time for this 20 milligram per ml Dextran concentrations. This indicates that, and these things will show fluorescence when they come and interact with the cell surface. So all this indicates that this result indicates that their diffusion phenomena is usually the hopping phenomena, where one cell will transfer the enzymes to its neighboring cells. This is what they have checked in this experiment. And from the previous experiment, they identified that with increasing concentrations, With increase in concentrations, they are able to reduce this mobility of the enzymes on the cell surface. That can be seen there here, that we can see that the diffusion coefficients goes down. This is the mobility based experiment where we can see that for the PBS the fluorescence recovery is highest, but for this Dextran with 320 milligram per ml the fluorescence recovery is lowest. And this is because the diffusion coefficients are this is because the diffusions of the enzymes at high concentration of macromolecular Crowder is lower. That can be seen here, and this is true for both the cell surface and bulk. So together they conclude that with increasing the concentrations of the crowding macromolecular, they can reduce the mobility of the surface enzymes. At the same time, the diffusion and exchange of enzymes also goes down. So it can focus directly only on the cell surface reactions. That's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all from this video. Thank you for your time.